Right now, as you listen to this, there are over 1 billion people on Earth walking around with these pressurized, fluid-filled biological tubes secretly thrashing inside their bodies.
Yeah, it is a staggering number to think about.
It really is. And, when you hear the word worm, you probably picture something very different. Like, you imagine this gentle, segmented earthworm wriggling on the sidewalk after a spring rainstorm.
Right, something totally harmless.
Exactly. It's this familiar, almost comforting image of nature. You expect a worm to just be a worm. But today, we are taking that comforting image and completely turning it inside out.
Oh, absolutely, because the reality of what a worm actually is, especially when we look at the ones that infect humans, is vastly more complex and honestly incredibly alien.
Yeah.
It's just a lot more impactful to human history than most people realize. We use this folk category of worms in everyday language, just, lumping everything together.
Like a worm is a worm.
Yeah, but if someone has a parasitic infection, the public often conflates tapeworms and roundworms as if they are basically the same organism. And biologically, anatomically, medically, they belong to entirely different phyla of animals.
Okay, let's unpack this because the stakes here are massive. We are diving into organisms that literally blanket the globe. Our mission for today's deep dive is to explore the hidden world of roundworms, which are also known as nematodes.
Yes, nematodes.
And to do this, we've gathered this huge stack of sources. We've got historical public health archives, taxonomic research, clinical parasitology texts. We're going to figure out how these organisms shaped our immune systems, dissect the massive public health campaigns designed to wipe them out, and look at the absolute cutting edge of biological research.
It's got a lot of ground to cover.
It is. But first, let's establish why a tapeworm and a roundworm are so different.
Well, to say those two are the same just because they are both, long and squiggly is like saying a snake and an eel are the exact same animal.
Oh, wow. That different.
Totally that different. Tapeworms are flatworms. They belong to the phylum Platyhelminthes. They have a completely different body plan, and they often lack a complete digestive tract entirely. They literally just absorb nutrients through their skin.
Oh, weird.
Yeah, but nematodes, or roundworms, they belong to the phylum Nematoda. The diseases they cause, their life cycles, and the specific mechanisms of the drugs we use to treat them, they share almost zero overlap with flatworms.
Okay, so before we get into the bizarre ways these organisms infect us, we really need to establish what a nematode actually is. Looking at the basic biology in our sources, it's essentially a pressurized tube.
That's exactly what it is.
Yeah, like if you strip away the clinical jargon, they are unsegmented, they are cylindrical, and unlike that earthworm on the sidewalk, they don't have a true body cavity.
Right. They possess what is known in biology as a pseudocoelom.
A pseudocoelom.
Yes. So a true body cavity, a coelom, is fully lined with specialized tissue called mesoderm. Nematodes just lack that complete lining. Instead, their internal fluid cavity is under really high pressure, and it's tightly packed inside a very tough, protective outer layer called a cuticle.
So essentially, a roundworm is functionally a biological water balloon, or like a hydraulic tube.
That is a highly accurate way to visualize it, yeah. The cuticle acts as an exoskeleton, and the pressurized fluid inside acts as a hydraulic skeleton, which keeps the worm completely rigid.
Right.
And because of this architectural setup, they only possess longitudinal muscles.
Meaning they run lengthwise.
Exactly. Muscle fibers running strictly down the length of their bodies. They do not have circular muscles wrapping around them like earthworms do.
Which perfectly explains why they move in such a disturbing way. I mean, an earthworm can stretch out and contract its circular muscles to gently inch along the ground.
Yes, very coordinated.
But because nematodes only have those lengthwise muscles, they can't flex up and down and they can't crawl. So if you watch them under a microscope, they just violently whip and thrash from side to side.
It's violent, yeah. And trapped inside that rigid, pressurized cuticle, they run into a severe mechanical problem when they need to grow. The cuticle just does not stretch indefinitely.
So they get stuck.
Right. So to get bigger, the nematode has to molt. It periodically sheds its entire outer layer, typically 4 times across its larval stages, before it reaches adulthood.
Wow.
And that highly specific biological mechanism, molting, is what triggered one of the most explosive taxonomic upheavals in modern zoology.
Yeah, this part of the research completely blew my mind. For most of the 20th century, biology textbooks universally grouped nematodes with earthworms and mollusks. Scientists categorize animals based on their physical shape and their body cavity architecture.
If it looks like a worm, put it in the worm bin.
Exactly, a worm went in worm bin.
And scientists who classify animals by their physical structure are called morphologists. For decades, traditional morphologists considered grouping by body cavities to be completely settled science.
Right.
But then, molecular phylogenetics arrived and dismantled that entire system. In 1997, a team of researchers, Anna Marie Aguinaldo, Rob DeSalle, James Lake, and their colleagues, decided to just look past the physical shape and sequence the organism's ribosomal RNA.
Now, how does sequencing RNA actually change the family tree? Like, what does that show us?
Well, ribosomal RNA acts like a molecular clock. Because the ribosome is so fundamental to life, it is literally the factory that makes proteins in cells. Its genetic code mutates at a very slow, very predictable rate over millions of years.
Okay, follow.
So by comparing the RNA sequences of different animals, you aren't just comparing what they look like on the outside, you are reading their fundamental genetic barcode to see exactly when their evolutionary paths split.
And when they read the barcode for the nematode, it didn't match the earthworm at all, right?
Not even close. The RNA sequence data prove that nematodes actually share a much more recent common ancestor with arthropods. We are talking about insects, crustaceans, spiders, millipedes.
Wait, really? So a microscopic parasitic worm is genetically closer to a crab than it is to a garden earthworm?
It is. And what's fascinating here is how the physical trait we discussed earlier, the molting, suddenly made perfect evolutionary sense. Earthworms don't molt. But spiders, crabs, and insects all undergo a process called ectotysis, where they shed an exoskeleton to grow.
Oh, of course.
Yeah, the genetic data revealed that nematodes share this exact same ancient mechanism. This discovery led to the creation of a massive new biological supergroup called Ecdysozoa. It forced this painful, paradigm-shifting reclassification that completely shattered the older assumptions of zoology.
That is wild.
Yeah.
But this struggle to simply categorize these organisms, it wasn't just some abstract academic drama. The inability to properly understand what these worms were and how they functioned had massive real-world consequences for medical history and human health.
Oh, absolutely. If we look back at the early 1800s, European medicine was operating under Carl Linnaeus's messy classification system. Linnaeus had this catch-all bin he called vermes, where he just threw in leeches, flatworms, roundworms, and even random mollusks.
Just a junk drawer of squiggly things.
Basically, yeah. But a Swedish-German naturalist named Carl Asmund Rudolphi finally started breaking this up through careful anatomical dissections. He proved that nematodes had a complete gut, a distinct mouth-to-anus tract, whereas flatworms had a blind gut, where food goes in and waste comes out the exact same opening.
Wow. But even with Rudolphi establishing that basic anatomical clarity back in 1808, the sheer scale of the public health impact wasn't fully grasped until a century later. Looking at the archives from the early 1900s brings us to an American zoologist named Charles Wardell Stiles.
Yes, Stiles. He was working for the U.S. Public Health Service, and he identified that a specific nematode, which was later named Necator americanus, was silently ravaging poor rural populations across the American South. We know it commonly as the hookworm.
Right, and this parasite was causing chronic lethargy, severe anemia, and stunted growth in children. But the really tragic part of this history is that society at the time didn't blame a microscopic parasite. They blamed the people suffering from the infection.
Yeah, it was awful.
The lethargy was popularly and very cruelly dismissed as an inherent cultural laziness.
But Stiles' discovery proved the lethargy was the direct result of a biological infection, which led to the Rockefeller Sanitary Commission campaign between 1909 and 1914. This was one of the first massive public health deworming efforts in U.S. history.
And we have to look at the dual nature of this campaign, because our sources highlight it as a really critical lesson in public health communication. Medically, it was an undeniable triumph. I mean, they treated hundreds of thousands of people and vastly improved physical health.
Definitely.
But the historical framing of the campaign was deeply troubling. When we look at this impartially, the public messaging often leaned heavily into those existing stigmas. It framed poor Southerners as unclean or inherently backwards.
And modern global health communicators still study this era as a cautionary tale, because the messaging masks the reality that hookworm is a soil-transmitted helminth. The lethargy was not a moral failing, it was a structural sanitation problem.
Exactly.
When a community lacks access to proper plumbing, human waste contaminates the soil. Hookworm larvae hatch in that soil, and they don't wait to be swallowed. They are heat-seeking.
Oh, that's terrifying.
Right. When a person walks barefoot over the dirt, the microscopic larvae use specialized enzymes to literally dissolve the proteins in the human skin, burrowing directly through the foot to enter the bloodstream. It was an infrastructure issue, completely divorced from a person's character.
Which perfectly transitions us into understanding how these microscopic thrashing tubes actually navigate the human body today. Our sources divide these into two main strategies, starting with direct cycles.
Direct cycles, yeah.
This means the nematode passes from human to human or environment to human without needing to develop inside another animal species first.
And the most prominent example of a direct cycle is Ascaris lumbricoides, the giant roundworm.
Giant is almost an understatement. These nematodes can grow up to 35 centimeters long.
Yeah, that's over a foot.
Just huge.
What?
And they currently infect roughly 1 billion people globally, primarily in regions that still struggle to fund sanitation infrastructure. A single female Ascaris can lay 200,000 eggs every single day.
It's an industrial scale of reproduction.
But the numbers aren't even the wildest part. The clinical texts detail this terrifying body tour that this worm takes once a human ingests the microscopic eggs from contaminated soil or food.
The mechanical journey is just astounding. You swallow the egg and it hatches in your small intestine. But the larva doesn't just stay there and grow.
No.
It uses enzymes to bore through the wall of your gut, entering the bloodstream.
Oh my God.
It rides the circulatory system until it reaches your lungs. Once in the microscopic air sacs of the lungs, the alveoli, it physically breaks through the tissue. It migrates up your windpipe, gets coughed up into your throat, and then you unconsciously swallow it back down. Only on this second trip into the digestive tract does it mature into an adult worm.
Okay, wait. I have to push back here.
Yeah.
From an evolutionary standpoint, that seems wildly inefficient. Why go through this totally complex, destructive lung migration just to end up back exactly where you started?
It's a great question, and biologists have debated this exact thing for decades.
That makes no sense.
Well, the leading theory is that this tissue migration is a deep evolutionary holdover. The ancestors of Ascaris likely infected humans by penetrating the skin, just like the hookworms we just talked about. If you enter through the foot, you must use the bloodstream and lungs to eventually navigate up and down to the gut. Even though modern Ascaris evolved to be swallowed directly, the ancient genetic programming for that tissue migration is still hardwired into its DNA.
That is fascinating.
Plus, the oxygen-rich environment of the lungs actually appears to be biologically necessary for that specific larval stage to trigger its next molt.
It is bizarre, and that lung breakout phase causes real harm. It often results in a feverish, wheezing illness called Loeffler syndrome.
Yes, it can be severe.
But before you feel too comfortable, just because you live in a place with pristine modern plumbing, we need to shatter a persistent myth. Nematodes are not just a tropical or poverty problem.
Not at all. Let's examine Enterobius vermicularis, which is the pinworm. Pinworms thrive in wealthy, highly sanitized, temperate countries. In fact, it is arguably the most common helminth infection in the United States, particularly concentrated in daycares and elementary schools.
And the mechanics of this infection are just the stuff of nightmares for parents. Because unlike Ascaris, pinworms live lower down in the gut. They don't do a lung tour. Instead, at night, when the host's body temperature drops slightly, the adult female pinworms migrate entirely out of the child's body.
Yep.
They crawl onto the perianal skin to lay their eggs.
And they secrete a sticky substance along with the eggs, which triggers an intense, localized inflammatory response.
In other words, severe itching.
Extreme itching.
The child scratches in their sleep, unknowingly packing thousands of microscopic eggs under their fingernails. The next morning, they touch their toys, their friends, or their own mouth, and the direct cycle continues. Because transmission relies on close human contact and shared surfaces rather than contaminated soil, a $1 million sewer system does absolutely nothing to stop it.
Which brings us to one of the most famous and frankly unglamorous diagnostic tools in pediatric medicine.
Yes.
The Scotch tape test. Because the eggs are deposited on the outside of the skin, a doctor or parent literally presses a piece of clear adhesive tape to the perianal area first thing in the morning. The tape lifts the eggs off the skin so they can be identified under a microscope.
Moving away from direct ingestion, our sources highlight the indirect cycles. This is where the nematode requires a vector like a middleman, an insect or crustacean, to develop before it can reach humans. Tackling these indirect cycles has led to some of the most ambitious global health campaigns in human history.
A prime example of this is Dracunculus metanensis, the guinea worm. The middleman in this cycle is a copepod, which is a tiny water flea that lives in stagnant drinking water sources. When a person drinks unfiltered water containing these infected copepods, the fleas are dissolved by stomach acid, releasing the nematode larvae into the host.
And then, about a year later, a meter-long adult female worm slowly and excruciatingly emerges through a blister in the skin, usually on the person's leg or foot.
It's intensely painful.
And when the blister touches water to relieve the burning, the worm violently expels millions of larvae back into the pond, and the cycle repeats. The traditional treatment, which goes back centuries, is to carefully wind the emerging worm around a small stick, pulling it out a few centimeters a day over weeks so the pressurized tube doesn't snap and cause a massive internal infection.
And some medical historians even argue that the ancient practice of winding the guinea worm on a stick is the true origin of the rod of Asclepius. You know, the universal symbol of medicine featuring a serpent wrapped around a staff.
Oh, wow, that makes perfect sense.
Mm-hmm.
But whether or not that's true, the modern story of guinea worm is a monumental triumph. In 1986, there were an estimated 3.5 million cases annually across Africa and Asia. Today, global cases are down to the double digits.
Incredible progress.
And they didn't achieve this with a miracle drug or a shiny new vaccine. It was driven entirely by behavior change and simple cloth water filters, spearheaded by organizations like the Carter Center and the WHO.
It really is poised to be only the second human disease ever eradicated following smallpox. However, the medical archives detail a massive last-mile complication. Eradication means 0 cases globally. Recently, field researchers were completely shocked to discover guinea worm hiding and reproducing in domestic dog populations.
Wait, dogs?
Yeah. For decades, the scientific consensus assumed humans were the only relevant host. This unexpected cross-species jump has significantly complicated the final push to true eradication.
Another devastating indirect infection is river blindness, which is caused by the nematode Onchocerca volvulus. The middleman here is a black fly that breeds in fast-flowing rivers.
And when the infected fly bites a human, it deposits microfilaria, these tiny larval worms. These larvae migrate freely through the host's skin tissue, causing maddening itching. Tragically, when these larvae migrate across the cornea of the eyes, the resulting scarring causes permanent blindness.
Now, the strategy to control river blindness hinges on a completely different approach than the water filters we just talked about for guinea worm. It turns on a single, incredible drug ivermectin.
Yes. The discovery of this compound from a soil bacterium in Japan by Satoshi Mora and its subsequent development for human use by William Campbell won them half of the 2015 Nobel Prize in Medicine.
So how does ivermectin actually kill the worm?
It is a highly targeted weapon. Ivermectin binds to specific chloride channels in the nerve and muscle cells of invertebrates. When the drug binds, it forces these channels open, flooding the cells with chloride ions. This essentially short-circuits the nematode's nervous system, causing complete muscle paralysis.
Oh, because it relies on that pressurized tube.
Exactly. The worm is trapped inside its rigid, pressurized cuticle, unable to feed or move, and it just starves to death. It's incredibly toxic to nematodes, but because human biology is structured differently, it is remarkably safe for us. Since 1987, the pharmaceutical company Merck has been donating ivermectin indefinitely to stop river blindness.
Here's where it gets really interesting, though. We cannot discuss ivermectin without addressing the massive cultural controversy from 2021. Looking at the public health archives, impartially, during the COVID-19 pandemic, unverified claims circulated that the drug could treat or prevent the virus.
Right. This led to a sudden surge in people purchasing veterinary formulations of ivermectin. These were highly concentrated pastes explicitly meant for deworming horses and cattle.
And it prompted the FDA to issue a widely shared warning saying, "You are not a horse, you are not a cow."
Yeah.
Again, looking at this impartially based on the history of the molecule, the irony is striking. You have a profoundly successful, Nobel-winning human medicine that works by paralyzing the muscle channels of parasitic worms, saving millions of people from permanent blindness. And suddenly it becomes culturally entangled in internet memes and medical misinformation regarding an entirely unrelated respiratory virus.
It really illustrates how easily the rigorous, mechanistic biology of a life-saving drug can be completely overshadowed by a viral reputation. Understanding the actual science of these antiparasitics is critical so we don't lose sight of their intended miraculous functions.
Which brings us to the final and undoubtedly the most bizarre frontier we found in our sources. For over a century, humanity has invented paralytic drugs, built vast sanitation systems, and handed out millions of water filters to eradicate these thrashing tubes from our bodies. Yet there is an active, ongoing debate in the medical community about whether we should intentionally swallow them.
Yes, you were referring to helminthic therapy.
Yeah.
This entire concept stems from the hygiene hypothesis. The core idea is that the human immune system did not evolve in a sterile, modern vacuum. It evolved over millions of years, locked in a biological arms race with parasitic worms.
How exactly did worms shape our immune system?
Well, we have an entire branch of our immune response, the Th2 response, heavily utilizing specific white blood cells called eosinophils. For a long time, scientists knew eosinophils were part of the immune system, but weren't entirely sure of their primary role. They don't fight tiny viruses. Instead, eosinophils are highly specialized anti-parasite troops.
Okay, so they fight worms.
Right. When they encounter a massive nematode that is too large for a white blood cell to simply swallow and digest, the eosinophils surround the worm and dump toxic, highly acidic granules onto its cuticle to destroy it.
And the hygiene hypothesis suggests that because we live in industrialized, highly sanitized countries without these worms, this entire branch of our immune system is essentially unemployed. It's just bored.
That's one way to put it, yeah.
Without massive parasites to dump acid on, it starts misfiring and attacking our own healthy tissue, leading to a huge rise in autoimmune diseases and severe allergies.
And based on this hypothesis, a minority of researchers and patients are deliberately infecting themselves. They swallow specific doses of hookworm or whipworm eggs.
But how does swallowing a parasite actually stop an autoimmune disease like Crohn's or multiple sclerosis?
It basically comes down to chemical warfare. Nematodes want to survive inside you for years. To avoid being destroyed by those eosinophils, the worms actively secrete specialized immunosuppressive proteins. These chemicals essentially act like a local anesthetic for your immune system, putting it to sleep so the worm isn't evicted. The medical theory is that by introducing the worm, its immunosuppressive chemicals inadvertently calm down the host's rampant autoimmune inflammation.
So what does this all mean? Are we fundamentally too clean for our own good?
We must be incredibly cautious here. While some small clinical trials have shown modest immune-modulating effects, helminthic therapy remains highly experimental. It is completely unregulated in most places, and it is firmly rejected by mainstream treatment guidelines.
Right, because it's a parasite.
Exactly.
Yeah.
Deliberately introducing a parasite that evolved to drain your nutrients carries immense biological risks. It is a genuine, unresolved scientific debate, not a settled cure.
It is just a wild twist in the human-nematode relationship.
Yeah. And if we connect this to the bigger picture, you can see why this entire field of microbiology matters so much today. Nematode diseases sit directly at the intersection of poverty, agriculture, and global health. Treating soil-transmitted helminths is one of the most cost-effective public health interventions in existence, often costing just cents per child per year.
That's amazing.
But the landscape is actively shifting. Climate change is currently expanding the geographic ranges of the vectors, the black flies and mosquitoes that carry those indirect nematode infections. The footprint of these ancient diseases is not fixed, it is moving.
As we wrap up this deep dive, I want to leave you with one final, mind-blowing fact that we have completely held back until this exact moment.
Yeah.
Through this entire discussion, we have framed nematodes purely as parasites, invaders, disease causers. But not all roundworms are parasites.
Oh, they're not.
In fact, one of the most important animals in the entire history of modern science is a completely harmless microscopic soil nematode called Caenorhabditis elegans, or simply C. elegans.
Yes. In the 1960s, a biologist named Sidney Brenner was looking for the perfect model organism to study complex biology, and he chose this tiny, transparent worm.
And because it is a transparent pressurized tube, scientists were able to map the development of every single one of its 959 cells. They tracked exactly how a nervous system wires itself together from scratch. The study of this non-parasitic roundworm has led to multiple Nobel Prizes in genetics, developmental biology, and our fundamental understanding of how human cells undergo programmed death.
It is a profound biological dichotomy. The exact same anatomical architecture, the cuticle, the pressurized fluid, the longitudinal muscles that makes Ascaris and guinea worms such devastating human pathogens, is exactly what makes C. elegans the perfect lens through which to understand our own biology.
It really makes you rethink that image we started with, the expectation of a simple, comforting earthworm on the sidewalk. Because the biological reality is infinitely more complex. The exact same phylum of thrashing, molting, fluid-filled tubes that causes some of humanity's most ancient diseases. Well, it also holds the foundational secrets to human genetics. Something to think about the next time you hear the word worm.